Okay, good day everyone, and thank you for joining today's webinar. My name is Joseph Richardson Larby, and I am the host and moderator of today's webinar, which is titled Productivity Improvements for Cost-Effective CER Writing and Maintenance. Our guest speaker is Ms. Shruti Sharma, who I will introduce soon. Okay, so to run you through today's agenda very briefly, there will be an introduction to Celegence, and then we go into the EU MDR's PMS and vigilance reporting requirements, key challenges and solutions, and then we will have a CAPTIS demonstration by Ms. Shruti Sharma, and she will also take us through a case study, and we'll have a Q&A session at the end. So, introduction about Celegence. Celegence is headquartered in Chicago, Illinois. We also have offices in the UK and in India, Bangalore. Celegence is a consultancy firm, and our mission is to provide regulatory value-added services for life sciences companies. And we prize that on three main pillars. So that is to improve compliance, increase efficiency and productivity, and most importantly, to reduce cost. A few of our clients or partners, as I would like to refer to them, can be seen on the right-hand side of the screen. So right from Novartis, Johnson & Johnson, and etc. At Celegence, some of our capabilities include MDR gap analysis, medical writing, so that is your clinical evaluation performance, clinical evaluation plans, clinical evaluation reports, and post-marketing clinical follow-up plans and reports, amongst others. So other regulatory intelligence consulting, technical file remediation, etc. In terms of medical guidelines and regions that we cover all regions across the globe. And for the therapeutic areas, from respiratory, radiology, urology, etc. So you can get in touch with us in all sort of therapeutic areas. We've got experienced consultants on board, or subject matter experts that we use, and in combination we hold over 300 years experience when we combine the experience of our SMEs. Okay, so to introduce you to today's speaker, today's speaker is Ms. Shruti Sharma. She is the senior medical writer at Celegence. She's actually my go-to when I need advice in key aspects when it comes to CEPs and CERs. Ms. Shruti Sharma has got 5 years of experience in medical writing. So that is with your clinical evaluation plans, your clinical evaluation reports, and other post-market surveillance reports. So Ms. Shruti Sharma is very abreast with this topic, and she's the right person to take us through today's webinar. So without further ado, I will introduce Ms. Shruti Sharma.
Thank you, Joseph. Now, when I started doing CERs about 5 years ago, I mistakenly thought that I would get to spend a lot of time reading a lot of new innovative research and maybe get to analyze all sorts of data to write in my report. What I did not know was that I'd be spending a lot of time collecting data and maintaining Excel sheets, tracking my data, making sure everything's recorded correctly in a safe place. If you or your team also spend a considerable amount of time on Excel sheets, something like this, then this webinar is definitely for you. Today's session, we'll cover a brief overview of the PMS reporting requirements under the EU MDR. We'll understand some pain points that writers like me experience when we create and maintain these reports. We'll also learn to leverage technology to automate tasks and save our time and cost. And also, I'll give you a brief demo of CAPTIS, which saves 20% of our team's time. Here we see a gist of the PMS reporting requirements under the EU MDR. We have the clinical evaluation report, PMCF, SSCPs, risk management, we have PSURs, PMSRs. Now, we can do entire webinars on each of these reports, but that's not what this webinar is for. What I want to highlight is that these are the typical reports a manufacturer may need for one device. Now, most of these reports have to be created for every device on the manufacturer's portfolio, and all of them may have overlapping and redundant data. Now, let's take a look at all that goes into these reports. On our left, we see a list of all our internal data sources. That is, we have all our manufacturer held data. This can include your device information. We have claims, internal testings. We have risk documents, PMS, FMEAs, claims matrix, and all that stuff. On our right, we have all the data that we collect from external sources. That gives us all our peer-reviewed literature. We have multiple databases where we can get all this. We also have clinical trial registries and also adverse events databases. Now remember, all of this data goes into documentation for one device, that too for one cycle of these reports. These reports have to be maintained. That means you have to repeat the process of data collection and analysis all over again when it's time to update these reports. Manufacturers must invest time and resources to execute these. And if you're only relying on traditional manual means to collect, you would have to deploy a lot of resources to meet your timelines. This could be a big challenge for all companies that cannot continuously add resources to their teams. I want to touch upon the problems and pain points that could arise when we manually handle all our data sources. Let's start with data collection. Right. So the biggest challenge with manual data prep is that one or more people must take time out from their day to gather data to use in any form of report. This could also mean that they are taking time away from analyzing this data and making an informed and timely decision. One would have to spend time devising some sort of a tracking system, maybe an Excel sheet like something we saw earlier, or perhaps a pen and paper system if that's what you're comfortable with. And there's often no central repository for all this information. Manual data in itself is tremendously time consuming and not really a value added activity for someone like a PhD or a clinician in your team. The time and expertise of these highly skilled resources should be targeted towards actual data analysis versus collection or standardization of data. Now let's talk about consistency. If your data isn't widely available, or if your business logic itself isn't centralized, your teams are reporting on different and conflicting data sets. One department or user may capture and calculate something one way while another calculates it entirely differently. This lack of a single source of truth can lead to distrust in your actual data, and it usually leads to more manual manipulation at a later point in time. Over time, this can lead to data silos. That means it becomes harder for cross-departmental agreement and collaboration. Problems with consistency could come from, for example, the way people record their search strategies, or maybe how you capture your article details. Your templates could have inconsistency, or maybe a strategy change in your search protocol itself. And we know this happens often. And writers have to spend additional time collating all this additional data and trying to make it look uniform so that it fits into your original data set. You can also have problems with reporting now. So your manual processes would inevitably mean inaccuracies or errors in calculations and reporting. Simple mistakes like a copy-paste error, or maybe you put in a wrong VLOOKUP formula in your Excel sheet. These compound over time, and these can go unnoticed before it's too late. Additionally, there can be problems with article traceability, referencing, and variations in the report output from, say, one user to another. Coming to maintenance now, you remember all the reports that we saw earlier. Now, all of those have to be maintained periodically. For us to have consistency with the initial report, we need to ensure that we have everything that we need right from the original plan. which means you need to have your older search strategy. You need to have all of your previous screening and appraisal data, all your previous referenced articles, or your source documents from your first report. And if you have a lot of individual contributors, some of them may not be permanent employees, their methodology or standardization may be different from what you follow internally. And if all of their work is not really stored internally in a safe place, then that means all of that raw data could be a challenge when it finally comes to maintaining these reports at a later point in time. Now, what if there was a solution to all of these challenges while also improving your team productivity, report quality, and also help you with compliance? What if technology could help you with bulk import of search data, maybe automatic import of free full text, article audit trails, give you customizable data collection forms, also give you a centralized repository for all of your reusable data, help you with data modularization. Now, this is important because one manufacturer may have different devices which pretty much follow the same, say, therapeutic area, and also the disease conditions that they treat are also similar. They can leverage data from one report to another, maybe content like your clinical background, your state of the art, some of the sections from the state of the art, that is maybe your guideline section, something about the alternative therapies. All of this can be leveraged for other devices also, as long as they fall under the same therapeutic area. Now, what if technology could also help you optimize your entire data collection and storage process, and also give you automatic triggers for report updates? The answer is yes, it can help you with all of that, and it has. I want to introduce all of you to CAPTIS. CAPTIS is a proprietary technology that we've built that allows our team to reduce manual effort and produce higher quality CERs in a lesser amount of time. A few of the features are listed on the slides, but the goal is to have one place where the writer processes both internal and external data sources in order to create the final report. Now, since MDR requires the various reports to be updated on a regular basis, you'll always have a high level of effort going into maintenance of these reports. By working with Celegence and getting access to this solution, you can seamlessly maintain your reports on a regular cadence going forward. Let me show you CAPTIS in action now. So this is CAPTIS. This is your reports overview page. This is where you'll get to see a brief summary of what's happening in your project. You'll get to see if you have any pending actions against all the articles that you retrieve for your DUE searches or for your state-of-the-art searches. We also have a little notice board at the bottom where you can leave comments for yourself. Maybe you need to follow up with someone, or if you have any collaborators on this project, they can also leave comments for you. Let's go to the search homepage. This is where all of your searches get stored. We see a couple of searches that I've run already. To conduct a new search, you click on the search option here, and we see a couple of databases listed here. Now, PubMed, Europe PMC, and Google Scholar are directly integrated with CAPTIS. That means any searches that you do on these platforms, you can conduct them on CAPTIS directly. The next three databases, that's Embase, Cochrane, and Prospero, you can search the parent websites and import all your article details into CAPTIS. Now let's run a sample search for PubMed. So the first data field that we see here is something called a search name, where you can assign a name to your search, whether if you're doing a particular string for your clinical background, maybe you're doing a search for your alternatives, all of them can be captured here. Let me show you how. Right. So we have our search strings here, and what you see in black and in bold, like competitor, alternative therapies, or clinical background, you can capture all your search objectives here itself. Meaning, because you have multiple strings within the same project, it can get confusing if you can't remember why you chose a particular set of keywords or why you ran a particular string with a particular set of filters. You can assign names to your searches so that you can easily identify the objective. Let's go back to our search page. Let's do a very simple search. I have a very basic set of keywords with me here. I'm going to call it a test search. Let this be a search for our DOE. Now, CAPTIS allows you to set your preference. That means, do you want to do a search for your device under evaluation? Are you only looking for safety and performance data for your device? Or are you doing a search for state of the art? You can choose either of these two options from here. You can assign a search category. Because we're running a full string, we will assign it as scientific search. You can then set your date range. Let's just choose 1 year for now. You can even choose article types, just like you can on PubMed. Let's say we're only looking for clinical trials for now. You can even choose your languages, and CAPTIS also has a species filter, just like on PubMed. You can select this, and let's hit search. Right. We got 243 articles on CAPTIS. We can verify this. If I go here, this will expand the string like the way PubMed expands the string. And then I can hit the globe icon here. It will take me to my PubMed page. We see the same number of articles. Now, when you've run your search, you can quickly browse through your results to see if these look relevant to your search objective. And then if they do, you can simply hit save here, and CAPTIS will save your search. Let's go back to our screening homepage. Right. So this is the string that we just ran, and it has captured all our details. Now, if you look closely, CAPTIS has also captured which filters were used when you were running this search. It has captured the string, obviously, and we also see which search objective was it that the search was conducted for. We also see which database the search was conducted on and what time, what date range was used. So this is essentially your entire search strategy in one place. This really helps us with traceability of our articles, and it also ensures that we record the correct information on our search protocol in our CERs or any other report it is that you're writing. This will also eventually help you with your reviews. If your team performs your searches and their reviews on CAPTIS, a reviewer can just log on to this page and then see what kind of searches were run, what kind of search strings were used. They can go and verify the results also. To begin screening, let's go to our screening homepage. Let's say we're doing a SOTA screening. Let's go to level 1. So systematic literature reviews typically have 2 levels of screening. Level 1 is where we're looking at the title and the abstract only, and level 2 is where we look at the full text to make a decision. Right now, we're in level 1 of my report, and I have a couple of filters on my right here. Before we jump onto filters, now, because we have multiple search strings and also we have multiple databases, it's quite likely that we will have duplicates. CAPTIS automatically identifies these duplicates. Like right now, we have 52 articles that CAPTIS identified as duplicates, and these are automatically excluded for you so that you don't have to deduplicate anything on your own. Now let's select a couple of filters. Let's say I want articles from PubMed, and I want all my articles that I'm yet to review. Right, we see 536 articles here. To begin reviewing an article, you can simply click on it, and we see a new window pop up. On our right, we see all our article details. We have the article title here. We see our authors. We also have other details like the DOI, PMID, and all of these are clickable hyperlinks. You can click on any of these links, and it will take you to the original PubMed search. So you can see the abstract below. You can read the abstract, take a decision. If this looks something that you want in your report, you can simply hit include here. And that's it. Let's jump on to our next article. To exclude an article, you can simply click exclude here, and you get a drop-down list of exclusion reasons that you can choose. Now, this list is fully configurable, meaning you can add as many reasons as you want depending on the process or the template that you follow for your reports. Let's say this is a different indication, and I've excluded that. A really cool feature that I really like about CAPTIS is article tags. Now, let's say you're doing your state-of-the-art search and you know there are multiple objectives that you need to fulfill. That means you have a section for clinical background that you need to write. You also have alternatives that you need to cover. Now, I look at this article and I think, oh, this has some great content for my alternative discussion maybe. I can tag it for alternatives so that I remember later why I included this article. Now, say this article also has some great content that can be used for my clinical background. I can also assign another tag, clinical background, and we are good. Let's also include this one. Now, because we have two very distinct objectives in our search, that is the DUE search, that is for safety and performance, and this is device specific only, and the second objective being state of the art. Now, it's quite possible when you're doing a review for one objective and you come across articles which fulfill another objective. Say, let's assume this article has, now this is a SOTA review. Now, this article has content for my device. I will not include this for state of the art, rather I would include this for my device under evaluation. So I can exclude this article from this review using an appropriate reason, and I can actually push this off in my DUE search. CAPTIS will automatically push these articles into my DUE workflow, and I can review them as a part of my DUE set of articles. Other things that you can do is you can edit article details. Let's take another article. Let's assume you did not get your entire information that you were looking for. You need to edit some links. You can always go to the options here and go to edit details, and you can edit any article identifiers from here itself. Let's go back to our level 1. So here we see a couple of words highlighted or made bold in black, like the word brain here. Now these are keywords. Let's jump to our keywords filters below. Keywords are something that you can configure when you set up your project. So keywords is basically you telling CAPTIS to highlight something for you that you're interested in. So all the words that we see in red here are all our negative keywords, words that we don't want perhaps. These also act as identifiers for you to quickly identify whether an article looks like it could be relevant for your search or not. And all your blue words are all your positive keywords. So all the words in blue come up in bold and black like so. And all the keywords that you've highlighted for your negative keywords come up like so. Let's search for, say, hepatic. And let's also add a couple of other negative keywords. Right. So now that I know these keywords appear in the title itself, I know that this is not going to be relevant for my review. I don't even have to look at the abstract because the title itself tells me that this is something that I don't need in my review. So instead of going to the full article details, I can simply select all these articles from this page itself. You can select as many articles as you want. Then you can go to selection and update. CAPTIS allows you to do a bulk update on these articles. You can update either article tags if you want to add multiple tags to all of these articles. You can even remove any tags that you want removed from these articles. You can even do a bulk include or a bulk exclude. Let's say I want to exclude all of these articles because they are not in the indication that I'm interested in. You can select your reason and hit OK. CAPTIS will ask you for a confirmation. You hit OK. And there, all of them have been excluded from my list. Now, once you're done with your level 1 review, you can jump to your level 2 review. Before you even begin your level 2 review, it's important that you have the full text with you. Now, CAPTIS has a feature where it can automatically download all full text for you, provided that they are freely available. And for the ones for which it could not find the PDF, they come under this tag called missing articles. you can simply filter out all the articles which have a missing PDF, and then you can export this list out into an Excel sheet and you can send it to your purchase department. Now, remember those article tags that I showed you earlier? Right. So this becomes really helpful for you to do a quick focused search. That means, say, today I want to tackle my alternative section in my state-of-the-art review. I can always filter by this tag, and I see all the articles that I included for my alternative discussion. Then I can do a very focused review, having only this objective in my mind, that I need to screen for all my alternative therapies. So once I'm done with this set of articles, I move on to the next set of articles, which could be, say, my clinical background or my guidelines. Now, you can use tags in all kinds of ways. You can use tags to assign articles to certain people. You can assign tags to articles that you aren't really sure about. You need a second opinion. You can use this option. You can even configure specific disease states or, say, specific outcomes that you're interested in, and you want to categorize articles. So tags essentially works like buckets for you to categorize your information in. And the possibilities with these tags is pretty much endless. Now let's do a level 2 review. I'm going to filter by all my articles which have a PDF and which have an actions pending against them. Right. I see 2 articles here. Let's jump to one of these. So I see all my articles details again here. I click on this button here and this will open the full text for me. Let us also start our review simultaneously. Right. So on our left, we have the full text article. This window can be toggled into a larger view. So as and when you read the articles, you can start answering your level 2 questions. These questions were configured using the IMDRF guideline. This is optional. You can choose not to configure questions also. If you want a simple drop-down list like the way we have in our level 1, that is also something that you can do. Let me answer a couple of questions here. Other things that you can do on this stage is create your article summaries. Now, you can create your tabular summaries from this page itself, any data that you extract from these articles. All of these are free text boxes. Now, all of these boxes have also been configured by me when I was setting up this project. You can configure as many boxes or as many buckets of information as you want. Say, I'm just adding some sample text here. You can even add notes for yourself or add comments for another collaborator. Now, let's say you're reading one article and you come across some content which you think you can use in some other section of your CER. It means you want to highlight content in your article. Let's say this section has great content for my risk discussion in my CER. I can always highlight the section and I can assign it a tag. Maybe let's assign it risk. Let's also assign it a safety tag. You can add as many tags as you want. All of these tags are also configurable. You can add more custom tags in your project configuration. Hit OK, and that is saved. Now let us complete our review. Let's encrypt this, and we are good. So all article highlights that you make, all of these can be accessed here under snippets. Right. So here I see all my highlights that I made for any of my articles. You can filter these highlights to jump to a particular set. You can even go back to the parent article if you need more information on the highlights. That's also possible. CAPTIS also allows you to export your entire list to help you reference articles. That means I can filter all my included articles from my level 2. And I can export this list out into a reference manager. We have Zotero integration. You can use the same kind of export for EndNote also, or any other referencing software that you're using. Now, once you're done with your entire review, there are a couple of reports that CAPTIS automatically generates for you. Let's go to our SOTA reports. An example of these reports would be, say, your L1 exclusion summary. This gives you a list of all your exclusion reasons that you use, also the count of each of these exclusion reasons, and how many articles did you finally include after your L1 screening. Other examples of reports can be your L2 review summary, where you see all your article gradings and your entire appraisal here itself. All of these reports can be exported out into an Excel format. You also have something called an article data sheet, where you have all the data that you extracted from these articles. This is the article that we did together in this demo. Now, all of these column headings can be configured based on the kind of data that you want to extract from these articles. We also have our search parameters, which is essentially your entire search strategy. Now, this was automatically captured by CAPTIS when you were doing the search. That means there is no error in your entire search strategy in terms of capturing data. If you were to manually record this information in an Excel sheet or a Word document, it's quite possible that you miss out some filters perhaps, or maybe you miss out a parenthesis somewhere. And that could lead to errors in terms of repeatability of the search strategy. Now, because this software automatically captures all your details, you can be assured that there will be no errors in capturing any of these information. Now, all of your reports can be exported together using this button export, and we get to an Excel sheet. Let me quickly show you the Excel sheet. So this is how the export looks like. You have your article flow in your first tab, which is basically how many articles came in, how many went out in which level, and how many were finally included in the CER. We also see our L1 exclusion summary that we saw earlier, and our L2 review summary, where we have all our gradings, we have all our article details in this column. Your article data sheet has all your extractions from each of these articles. CAPTIS automatically generates your appendix for you in case you want to use this information in your CER. This works like evidence that you did indeed conduct a systematic literature review, and you have details from all the articles that came up from your search. You have all your author, title, source. You also have the decisions for each of these articles. Now, all of these articles right now say no because by default, all of them are no, as in when we take an action against them, CAPTIS will update the reasons for us. Like article number 18 was an exclusion, and this was the reason that we used. So all of this data can be copied from this Excel sheet into a table in your Word document. You also see the search parameters here, all the strings that were used, all the filters that were used for each of these strings. We also get to see how many articles were included or excluded from the string, and we also see how many exclusion reasons were used. Now let's jump back to CAPTIS. Right. Now let's jump on to the next module that I have for you. You can upload all of your source documents on CAPTIS, and these have been pre-configured to match the kind of data that we collect for all of our regulatory documents. All of these are pre-configured categories. You can drop information into each of these categories. What more you can do is you can add bookmarks. So you may come across a document, say a technical file, which is say 1,000 pages long, and there's a lot of information that you need from it, but all of it is jumbled in different pages. And you might maintain some sort of a tracker, maybe an Excel tracker, where you save which page had which information. On CAPTIS, you can do that using the bookmarks feature. Say this is a piece of information that I really need for my report. I can bookmark it. Let's bookmark all this content. Let's call it pre-operative. It's going to call it test for now. You can assign it a category. Which category of information does this fall in? Maybe this is your, let's say, product information. And this gets added into your list of bookmarks that you have. You can jump between pages, between bookmarks, and you can even jump between different documents. Now, that was just a small subset of features that we have on the platform. There's a lot more to show, and we can do that in a longer demo. You'll eventually be able to draft entire CERs on CAPTIS. And if you've created your initial CER on CAPTIS, creating a maintenance CER is pretty easy. This is how it look like. You can select sections which you want to carry forward to your newer documents based on any newer reports that you have and any new updates in your source documents. You can also set the date range for the new report. Because CAPTIS conducted your previous search, that means it already has all your search parameters. All you now have to do is put in the start and the end date of your maintenance report, and CAPTIS will automatically run these searches for you. You'll also be able to draft entire CERs on the platform, and this is how the drafts will look like. If you see the table of contents on the left, we see some of these sections have been marked in red. That means this is CAPTIS telling you that, hey, you did mention that you have updates in these sections because you have newer documents in these sections. So CAPTIS is giving you a prompt that you must update these sections based on the newer documents that you've uploaded. On the right, we see all our reports from our literature workflow. So any reports which are created in the literature workflow can be used directly into your reports in CAPTIS. You'll also be able to highlight and comment in case you're collaborating with multiple people here. Here we have a case study of an MDD to MDR remediation done through CAPTIS. Now, the client of ours was a manufacturer of a standalone software, which was used for imaging and visualization of cardiac anatomical structures. Now, the challenge that we had, now this was their first MDR CER that we were doing for them. And this had overlapping date ranges with the previous submission that was done on CAPTIS. Now, this device was widely used. It had been out for decades and a really popular device. And we know what that means. A popular device usually means we have a lot of data to sort through, which is not necessarily a bad thing in terms of evidence. It's just a lot of effort sorting through a lot of data. So we had about 1,000 articles reported for the one device. And let's see what we did. Now, because the previous MDD literature review was conducted on CAPTIS, and because the date ranges were overlapping, our writers could leverage screening decisions for over 800 articles. And we only had to screen about 150 of them, which were published in the gap period. That means the search actually gave us 950 articles, and we only ended up screening about 150 of them this year. Now, 950 versus 150, that is a significant difference in the number of articles to review and also the effort required to screen all these articles. Our medical writing team could save over 120 hours of effort in literature retrieval and screening. That means we had to deploy lesser resources to do the same project. This is just one of the examples of the benefits you get when you leverage technologies in your report. Imagine the cumulative savings in total effort and overall cost when you utilize technology for all of your maintenance CERs. Now, we have other customers who are using it for their own writers, and they get the same benefit that our team has been getting for the past 2 years. There are different ways you can utilize CAPTIS. Now, we offer an in-house model where your team utilizes CAPTIS for all of your in-house medical writing needs. You can also have a collaborative model where your team and our medical writers collaborate together on projects and communicate via CAPTIS. Or you can outsource your entire medical writing projects where our medical writers can create your reports on your behalf, and you will get full visibility and review options on the platform. With that, we come to the end of our webinar. Thank you so much for joining me today. If you want more information on CAPTIS, or if you have any questions on harmonizing your current process to the platform, feel free to get in touch, and our team will be happy to set up an in-depth demo with you. Thank you.
Thank you, Ms. Shruti. Thanks for taking us through the CAPTIS demo and its benefits and all the challenges. It is very much appreciated. We will go into a Q&A session and try and answer a few questions there. Okay. So I would like to ask Ms. Shruti Sharma to turn on the video.
Hi, Joseph.
Hi, Shruti. Thanks for joining again. That was a fantastic webinar and presentation there. Thank you. Very insightful.
Thank you.
Thanks. Okay, so the first question that's come through is, can I configure my own questions for exclusions? Sorry, you're on mute.
My bad. Yes. So is the question on custom exclusion reasons, is it, for CAPTIS?
Yes, so configuring their own questions for exclusions. So how they can configure questions in CAPTIS.
I wonder if I can share my screen. Right, I don't think I can. Yes, but you can configure all your own questions. Whenever you set up your project the first time on CAPTIS, you can configure the list of questions that you want for your level 1 and your level 2 also. CAPTIS also allows you to set templates, meaning if multiple CERs or multiple literature reviews follow the same exclusion reasons, you can set the template just once. And every time you start a new project, you can load the template so that you don't have to enter those questions anymore.
Okay, all right. Thank you. Thank you very much. And the next question is, can I reuse the state-of-the-art literature from one project to another?
Yes, if you conducted your state-of-the-art literature review on CAPTIS, you can actually clone an entire project. Meaning, say, we have device A for which we've finished our state-of-the-art review, and then we have device B, which is also from the same therapeutic area, which may need some content from the other state-of-the-art for the other device. You can clone project A, and then you can edit the date range. And all the answers that you have taken so far, all of them will still remain, and the gap, all the extra articles which came up in the gap date range, those will be updated as pending articles. So yes, you can leverage the whole search.
Okay, all right, thanks. And the next question is, how many people can access a project at once within CAPTIS?
Right, so CAPTIS was designed with collaboration in mind. We want cross-functional teams to be able to use the platform at the same time and also exchange information. That is why we haven't followed a user-based subscription model. Rather, it's a project-based subscription model. So you can have any number of users access the same project on CAPTIS. I'll be happy to discuss more about the pricing. If anyone's interested, you can get on a call with us.
Okay, all right, thank you. I think we've got time for a couple more questions. Can I generate article summaries in CAPTIS?
Yes. So at the level 2 stage where we saw the entire PDF, you can set up maybe summary tags, which are the same as your column headings for your tabular summaries. You can have text boxes which come up every time you view an article in full text in level 2, and you can summarize your article there itself.
Okay, all right, thank you. And the next one is, can I change my date range after I've performed my review?
You can. You can change the date range even after you've completed the entire review. So what happens is, say you have a date range of 5 years, and if you want to shorten it to 4 years, you can do that, and all your previous answers will still remain. And if you want to expand the date range, all your previous answers will still remain again, and you will only get new articles which came up in the gap search. So you will have to only take actions against the articles which came up in the gap search, the newer articles.
Okay, all right. Thank you very much. These are some of the questions that came through from the chat. But before we end today's session, Ms. Shruti Sharma, I would like to ask you one question. CAPTIS is, a new solution developed by Celegence at the moment. What would you say is just give us the one benefit of CAPTIS?
Right. So personally, I've used CAPTIS for a lot of my literature reviews. I was doing literature reviews even before technology came into the picture. So I've been doing the manual process, and I don't think I can ever go back to working on Excel sheets. The amount of efficiency using technology in general brings to your entire process. I don't have to, I know this is something that I've already covered in my presentation, but the amount of time that I save by not having to manually look for data and not having to manually collate it, that is such a blessing. So I don't think I'll ever go back to a manual search process after using a platform like that. And the fact that I can also have separate workflows for my state-of-the-art and my normal safety and performance searches. And even if I want to shift articles between both of these workflows, it's so easy. I don't have to keep track of any data anymore. So I think that is, those are my favorite features.
Goody. Well, thank you very much, Ms. Shruti Sharma. Well, guys, you heard it from the expert, and all the benefits of using CAPTIS to make medical writing easier. If you do have any other questions, or you would like to arrange an extended demonstration, please, the details are on the screen. Reach out to us, and we'll be able to configure something and arrange a demonstration and go in depth into the benefits of CAPTIS. So thank you for joining us this afternoon, and we'll catch you on the next one.
Yes.
Thank you.
Before we close, Joseph, I think we have a couple of questions on the chat. I see a couple. There was one question asking if our previous literature search was not conducted on CAPTIS, can we manage the overlapping dates in that case? So even if your previous MDD search was not on CAPTIS, you can be able to import the entire review on CAPTIS. That way, that's basically, you can export your literature review to CAPTIS. So that is possible. And other questions we got was, right, there was one question on screening about why did we exclude literature after the first screening. So systematic literature reviews, like I said, are typically in 2 stages. You have level 1 and level 2. Level 1 is where you're only looking at the title and the abstract and making a call whether something looks relevant to you or not. And you would not want to look at the full text of all the articles, because that would take up a lot of time and that's not really efficient. Doing 2 levels of review help you get to your final list of included articles faster, and also it's more efficient. And the last question that we had was, can I modify tags mid-project? Yes, you can. So even if you've configured the project, you can always go back to your settings page and make any more changes. Meaning any new exclusion reasons you want to add, you can do those too. And any new project tags, any new highlights you want to make, you can do everything. But if you change any existing reason, a reason that's already been used as exclusion for one of these articles, those articles would go back to the pending stage, just to make sure that we don't have errors in the final report that we generate. That's why CAPTIS ensures that we go back to the pending stage for any reason that has been edited. Right, I think that is all, Joseph.
That's it. Okay, thank you very much, and thank you everyone for joining today's webinar. Have a good day.